everyone, my name is Vincent, and today we're going to talk about phylogenetic tree construction. And it could be one of the question in year 12 exam. So in these questions, we're looking at from sequencing data, how do we construct a phylogenetic trees to represent the evolutionary relationship between these closely related primates of a human, chimpanzee, gorilla, orangutan, and a siamang. So the thing we're going to have to firstly look at is their given sequences. And the sequence has already been aligned through a classical program that is online available. If you're ever interested, you can play with it. But the alignment results is given as in this form. So if we look at them closely, we might find the first base pair is all A, so which is a good alignment, and second base pair is also all an A, which is no difference. Third T is okay, and the fourth base pair starts to observe differences between orangutan with the rest of the primates, and that is a point mutation to mutating these C into A in orangutan lineage. So therefore, it's the same for the next T. A point mutation occurs, and the next base pair aligned, no change at all. And then we're looking at something very different, as in the nature of the mutation there. So for human, chimpanzee, gorilla, and orangutan, there is a dash which is simply represent that base pair is not there. And for siamangs, there's an insertion of a G in that position. And the next base pair is all aligned as an A and a C. Again, for the next base pair, we see point mutations in chimpanzees, gorillas, and orangutans. And that base pair is not there for siamangs. So if we look at the last base pair, also there's two divisions in gorilla and orangutan. So given that information, how do we really construct that sequence into a phylogenetic tree diagram? Now, the method I'm going to introduce is called unweighted pair group method with arithmetic means, in short, UPGMA. Now, if we're looking at that, we would really want to know what are the differences between each of the group. So these differences can be arbitrary, but it can also be arbitrary, or it can be numerified from sequencing data. So if we're looking at it, these four sequences, we're going to choose human as a starting point, and it doesn't really matter which one you're going to choose as a starting point, but at least we need a starting point. So now we're looking at human, chimpanzee, gorilla, orangutan, and siamangs. So this is a matrix that annotates the differences between these these five animals. So human, chimpanzee, gorilla, orangutan, and siamangs. And we're going to construct this difference matrix to represent their distance towards one another. Okay, now if we're firstly looking at human, when human compared to himself, all the sequences are aligned, so therefore there's no difference, i.e. the difference score will be 0. Now then we're looking at a human and chimpanzee, which is these two groups. We're looking at basically all of the sequences that are aligned with each other. The only difference comes in the penultimate A and G. So for any point mutation, we're going to arbitrarily give it a score of 1. Point mutation and score of +1. And for an indel mutation, we're going to give a score of +2. That's just arbitrary. You can give whatever the score you want to, but based on scientific convention, that's generally the score we're giving. So when we compare it to human and chimpanzee, and we've already set it, and it's +1 in the point mutation score, so therefore we're going to do +1 in here. And now we're comparing human to gorilla, and human to gorilla in there, and this is +1, and that's +2, so +3 in total. And then human to orangutan, we have +1 and a 2 and a 3 and a 4 and a 5 and 6 and 7. So difference score between human and orangutan is +7. And human to salmons, and we're looking at indel event right there, so that's +2. And another indel event right there, that's +2 again. So +2, which is +4. Now, we've done a human to everything else comparison. We're just going to do pairwise comparison for all of them. So now we're looking at a chimpanzee to chimpanzee, which is 0, and so therefore the diagonal line is always going to be 0, and we wouldn't bother to compare them at all. So chimpanzee to gorilla, if we do that comparison right there, that's the same. Everything up to the A is the same, and in fact the A is the same as well. So G and nothing is +2 in score. So here we had given +2. And chimpanzee to orangutan, that's the same, and until we see that point of mutation, which is +1, +2, and then same, and +2 again. So +2 will be +4 in that sense. And chimpanzee to salmon, again, we're going to also have to eyeball the smallest value, which is human orangutan and salmon to human orangutan. Human chimpanzee to orangutan and gorilla group, and which is 4. And also you found this to be 4, and there's no problem of choosing this to be starting point, but I'm just going to choose this as a starting point, and just to be clear. Now, if that's 4, so therefore the distance between these two taxons are going to be 4 total distance, and because that is also shared in between two lineage, so that's going to be 2 and a 2, which is now this point here, which is a human, chimpanzee, orangutan, and gorilla group. If we now have eliminated the differences between these guys and can wipe it off and to give it a 0 in score. Now, arithmetic mean difference between this group to salmons will be 4 + 7, which is 11, divided by 2, which is 5.5 as a new distance. Now again, from here, the salmons will be having a distance of 5.5. Now, so therefore, these distance will be 2.75 and 2.75 there. I hope that makes clear, but in a reality, what we are looking for is not the genetic distance, how much you accurately calculate them, and what is important is the topology of the graph or the shape of the phylogenetic tree.